Thank you so much for inviting me. I hope you can hear me because I don't want to give a talk for 25 minutes and no one can tell me that that's the case. So please stop me if you don't hear me.
We can hear you very well.
Excellent. Thank you. And I'm sorry I can't be there, but I'm there in spirit. So I know we're running behind, so I'll get started. So thank you very much for the invitation. Let's see, I'm trying to move my slides here. There. So these are my disclosures. So, myeloma, obviously, we've made lots of improvements. And as you can see on the left, with the addition of the quads now in first line, we have amazing responses in the first line of therapy. And that's translated to overall survival for our patients, but unfortunately, no cure just yet. But when you look at patients who've actually been treated with CD38 antibodies and they become refractory, we know from the Mammoth study that the overall survival is quite poor for these patients, and it's only 8 months. And it only worsens as you get more and more refractory. So as you become refractory to the PI, the IMiDs, and the anti-CD38s, that response continues to worsen. So this is where the BCMA treatments come in. I think they fit very nicely in the post-CD38 space. And so BCMA is a great target. Obviously, you heard about CD19 CAR Ts, but CD19 is not really expressed in the majority of plasma cells. So we need another target. BCMA is that target. It is expressed basically in all plasma cells, and the expression increases as the cell becomes more malignant, and the expression is highest in myeloma. It is minimally expressed in other tissues, which of course makes it a great target. BCMA is actually a very important receptor for myeloma cells. It is a receptor for the TNF superfamily, a receptor for APRIL and BAFF, and it plays a key role in B-cell maturation and differentiation. So again, a very important target for therapies. We now have 2 FDA-approved BCMA-targeted CAR T cells. We have idacaptogene viloxazil, or ide-cel, and ciltacabtagene autoleucel, or cilta-cel. And these are both FDA-approved in the United States with the same indication for adults with relapsed refractory multiple myeloma after 4+ prior lines of therapy, which include an IMiD, PI, and an anti-CD38 antibody. In the EU, they have a little bit more lenient approval with just 3 prior lines of therapy, including a PI, IMiD, and anti-CD38 antibody. So how did we get these 2 FDA-approved CAR Ts? The first is ide-cel, and that was based on the phase 2 KarMMa trial, which I'm showing you here. So I'll just remind you that ide-cel is a traditional second-generation CAR T cell with a single anti-BCMA single chain variable binding domain that is linked to a 4-1BB co-stimulatory domain and a CD3 zeta signaling domain. Patients had to have 3 prior lines of therapy, and previous exposure to an IMiD, PI, and anti-CD38 was required. As you can see here, patients were treated, 158 patients were screened, 140 leukapheresis, and 128 patients actually underwent treatment. And you can see there were 3 dose levels of 150, 300, and 450 million CAR T cells. This is the baseline demographics of the patients, very high-risk patients with a median of 6 prior lines of therapy, so very heavily pretreated. And as you can see at the bottom, their refractory status, the majority of patients, 94%, were refractory to an anti-CD38 antibody, which fits that really refractory population. This is the overall response, and to the very right, you see that the overall response for the entire population was 73%, with a complete response rate of 33%. If you look on the left, you see that based on dose level, it appears that the higher the dose level, the better the response. So the 450 appeared to do best with an overall response rate of 82% and complete response rate of 39%. I'll remind you that in this population that is being tested, the expectation, and for the previously FDA-approved products, the overall response rate is in the range of 20 to 30% with CR rates of 1 to 2%. So clearly significant improvement from that. These are the PFS and overall survival curves. If you concentrate on the green curves, that's the entire population. So the median PFS was 8.6 for all patients, and the median overall survival was 24.8 months for all patients. Cell dose and depth of response matters. So on the left, you see the progression-free survival by target dose, and you see that the patients who received 450 million cells, which is the green curve on the top, you see that the median PFS is 12.1 months. And similarly, if you look to the right, In the patients that achieved complete remission or stringent complete remission, which is the light blue curve on the very top, you see that they did the best with a duration of response of 20.2 months. In terms of adverse events, hematotoxicity is very common, as was described previously, but it's important to note that it is true grade 3-4 hematotoxicity that can last. So about almost half the patients in the KarMMa trial were still had grade 3-4 hematotoxicity even 2 months after the CAR T. So that, I think, is an important thing to remember and to keep in mind. Cytokine release syndrome was very common and was seen in 84% of the patients. The majority were grade 1-2, with only 5% of patients having grade 3 or higher. The median onset was quick. It was just 1 day. So that's important to compare to the next product that you will see. But it was very manageable. Neurotoxicity was seen in 18% of patients. Again, mostly grade 1-2, 4% had grade 3 or higher. The median onset was 2 days, so similar to the time around the timing of cytokine release syndrome. We now have some real world data. So this product now has been out for about 1.5 years. So this is a group of academic centers showing what their real world experiences, and you can see it's actually very nice to see that the median PFS in the real world was very similar to what we saw in the KarMMa trial, as was the overall survival, granted a very short median follow-up. But what was very interesting with this real world study was that 77% of the patients would not have been eligible for the KarMMa study. And so again, showing us that we can sort of expand beyond the clinical trial population and do it safely with good results. So what about cilta-cel? So cilta-cel was studied in the CARTITUDE-1 study, and this was, I remind you that cilta-cel, it's a little bit different. It's the same structure as ide-cel, a second generation CAR T, except that it has 2 BCMA binding domains. And we think that maybe the 2 binding domains confer avidity and perhaps make this a little more sensitive. So similar population, 3 prior lines of therapy, PI, IMiD, and anti-CD38 antibody. 113 patients were enrolled, 101 underwent lymphodepletion, and 97 patients ended up getting dosed. And I show you here in on the slide that the target dose was 0.75 times 10 to the 6 per kilogram, and the median administered dose was closer to that at 0.71 times 10 to the 6. This is a much lower dose than we saw with ide-cel. So this translates to about 50 million CAR T cells compared to 450 million CAR T cells with ide-cel. This is the demographics of the patients. And again, heavily pretreated population, very similar, also 6 median prior lines of therapy, and 88% triple refractory. And you see at the bottom, 99% of the patients were refractory to anti-CD38 antibodies. Response rates were quite impressive, with overall response rate of 98% and stringent complete remissions of 82%. The median PFS and overall survival have not been reached. This is an update of 27 months, and the 27-month PFS is 54.9% for all patients. And at the top, you see the patients that achieved stringent complete remission. That's even better at 64.2%. And the median overall survival has not been reached. And the 27-month overall survival is 70.4%. So appears quite durable, which is nice to see in this population. In terms of safety, again, you see the cytopenias with many, a high percentage of grade 3-4 hematotoxicity. If you look over to the right in the cytokine release syndrome, you see that the majority of patients have cytokine release, but mostly grade 1-2 with 5% grade 3 or higher. But look at the median time to onset. This is, it was 7 days compared to ide-cel, which was 1 day. So it goes to show that it's really important to pay attention to the pharmacokinetics of the products, because they can differ quite a bit in terms of when to expect your toxicities. Now, neurotoxicity was another differentiator here compared to ide-cel. If you look at the total, there was 20% neurotoxicity with 10% grade 3 or higher. If we look at just ICANS, the traditional ICANS definition for CAR Ts, this was very similar to ide-cel, with 16% of patients having ICANS and 2% grade 3 or higher, with the median onset of 8 days around the time of cytokine release syndrome, which is what we saw with ide-cel. However, with cilta-cel, we did see 12 patients that had this other neurotoxicity that was high grade, with 9 of them having grade 3 or higher. And median onset was very late, of 27 days. And some of these, 5 of these 12 patients actually had what we call motor neurocognitive disorders, very Parkinsonian-like symptoms that did not respond very well to therapy. So an important toxicity to remember. Unfortunately, unlike what you saw with lymphoma, we're not really seeing a plateauing of the curves, and the majority of patients continue to relapse. So there has been descriptions of patients relapsing with BCMA negative disease, and this is just a case report of a patient actually having biallelic BCMA loss. But luckily, this is very rare, and the majority of patients actually relapse with BCMA positive disease. So what can we do once, beyond sort of what we have now? Obviously, in myeloma, everything seems to work better when we do it earlier. So there are many trials now looking at earlier populations, including CARTITUDE-2 and KarMMa-2, which I'll show you in a second, some of the early results from CARTITUDE-2. KarMMa-3 is a randomized phase 3 trial of ide-cel versus standard of care with patients with 2 or 4 prior lines of therapy. And we just saw a press release that they have met their primary endpoint, which is progression-free survival. And so we hope to see that data at ASH. Frontline-wise, looking at patients with high-risk disease, there are studies ongoing looking at actually CAR-T therapy, even in the frontline setting. So looking at the data that we actually do have, we have 2 cohorts from CARTITUDE-2, and this is with cilta-cel, of course. So cohort A is for patients who had 1 to 3 prior lines of therapy and they require that you be refractory to lenalidomide. Cohort B is for the functionally high-risk patients. So these are patients who progressed 12 months after starting their initial therapy or after stem cell transplantation. And so here, cilta-cel was used as second-line therapy. So if you look at cohort A, these are the patients that had a median of 2 prior lines of therapy and were lenalidomide refractory. You see that the overall response is 95%. So all but 1 patient responded with a complete response rate of 90%. With a median follow-up of 17.1 months, the 15-month progression-free survival is 70%. And the 16 patients that were MRD evaluable, all were MRD negative. So again, very deep responses. And so hopefully this will translate to prolonged responses. In terms of safety, very similar to what we saw with CARTITUDE-1. So hematotoxicity was important. Cytokine release was seen in 95% of patients, with 10% having grade 3 or higher. And the median time to CRS, 7 days. So again, similar to CARTITUDE-1. Neurotoxicity-wise, there was some neurotoxicity, but mostly grade 1, 2, median time to onset 8. So the more traditional ICANS, and they did not see any new cases of movement and neurocognitive neurotoxicity, which is reassuring. If you look at this cohort B, and this is the cohort that was functionally high risk, so patients who relapsed within 12 months of their initial therapy. So this was given a second line of therapy. You see that they have a 100% overall response rate with 90% complete response rates, with a median follow-up of 13.4 months. The 12-month PFS is 89.5%, so that's looking quite good, right? These patients all only had 1 year of duration of response to their initial therapy. So maybe we're starting to cross that line, which is a good thing to see. They had 15 patients that were MRD evaluable, and 93% were MRD negative at 10 to the -5. In terms of safety, again, very similar to what we saw with CARTITUDE-1. So hematotoxicity was the main toxicity. Cytokine release was seen in 84% of patients with grade 3 or higher, 5%, and median time onset of 8 days, again, similar to CARTITUDE-1. Neurotoxicity, the majority of patients who did have neurotoxicity were grade 1, 2 with grade 3 or higher of 5%, median time to onset of 11 days. but they did see one case of the movement neurocognitive AEs, and this was a patient that actually I spoke to the investigator that had this patient, and this was a patient that was had very high burden of disease, that had very refractory disease, and they could not get the disease under control with any bridging therapy. So they went into the treatment with rapidly progressive disease, and unfortunately, that is an increased risk of this late neurotoxicity. So what about, how can we make these better? One thing you could try and do is maybe increase the expression of BCMA. And so the group in University of Washington has done this with gamma secretase inhibitors. So gamma secretase actually cleaves BCMA on the surface of myeloma plasma cells. And so by using the inhibitor, you can increase the expression. And so they've used this by giving the gamma secretase inhibitor prior to a second generation CAR T cell. And so you can see here that on the left, they show very nicely that they can definitely increase the expression of BCMA. So you see the little blue bars compared to the orange. The blue is pre-GSI and the orange is post-GSI. So most every patient increased their expression or their BCMA surface density. And so on the right, you see the response rates, and you see that basically all but one patient has had a response, which is quite gratifying. And what's even more interesting is they actually have 4 patients who had prior BCMA CAR Ts, all actually had a response. So a nice sort of kind of proof of concept that perhaps you can augment the response to BCMA by using a gamma secretase inhibitor. One of the problems with this, at least early on, is that the gamma secretase inhibitor has some potential neurotoxicity. So it remains to be seen whether we can actually use that in combination with CAR T cells or the bispecifics. One of the problems with autologous CAR Ts, of course, is in a disease like myeloma, in the relapse refractory setting, is that these patients often are progressing and then have organ dysfunction. So perhaps an allogeneic CAR T would kind of do away with some of the problems of having to wait for the CAR T. And so we do now have some data from the Allo715 BCMA allogeneic CAR T, which uses a talented, so it has a traditional CAR, so second generation CAR infused, but then it uses talent technology to actually knock out the T cell receptor, which should minimize the risk of graft-versus-host disease. And actually, they have not seen any graft-versus-host disease. But the other thing they do is they actually knock out CD52. And so because of that, they're able to use an anti-CD52 antibody for lymphodepletion without hurting their CAR T cells. And so their lymphodepletion is a little bit different. They use fludarabine, cyclophosphamide, and the Allo647, which is this anti-CD52 antibody. And so the early data looks promising. And one of the things that is more impressive is that 90% of the patients were able to get treatment within 5 days of enrolling on the study. So lightning speed, which is nice. So off-the-shelf therapy, and the early response rates look promising. But again, more to come, I think, from this therapy. The other potential is, well, maybe if BCMA therapy is failing, perhaps we should go to another target. And definitely the next target that is the most advanced at this point in myeloma is GPRC5D. And you'll see a lot of data. And actually, hopefully, we're close to an approval of a GPRC5D directed bispecific antibody. But this is data with a GPRC5D targeted CAR T from Memorial Sloan Kettering with MCAR-H109. And so this is, again, a second generation CAR T. It's just that instead of BCMA, it's GPRC5D as the target. And as you can see, it is a dose escalation trial. They started at 25, went all the way to 450 million cells, and are seeing responses and all. But I'll point you to the table on the right lower corner of the slide, where they looked at patients who had prior BCMA therapy, and they had 10 of those patients, and you see an overall response rate of 80, and they had 8 patients who had prior BCMA CAR Ts and had a response rate of 75%. So again, this is reassuring that perhaps we can use a different target if and when BCMA CAR T or therapy fails. And then finally, from a standpoint of sort of kind of other things, manipulations that we could look for is perhaps to trying to minimize escape after one target? Could we do dual targeting? And this data has been shown. So there's a Chinese group looking at FastCAR GCO12F, which is a dual targeting BCMA CD19 CAR T. And so these patients, the usual relapse refractory after at least 3 prior lines of therapy, the patients went through the usual lymphodepletion and received the cells. And again, you can see that so far, at least early on, in 28 patients, we're seeing some impressive response rates in this population. The key here is going to be the durability in my mind. So, I think we're seeing the response rates are very high with a lot of these CAR Ts. The question is, are these manipulations going to be improve on the durability and hopefully eventually become curative? So the idea behind this dual CAR T is that even though CD19 is not really expressed in the majority of plasma cells, there is some data that perhaps CD19 is expressed in the plasma cell stem cell, and so the idea would be that BCMA would get you into remission, and maybe the CD19 CAR can prevent that relapse. And so again, only time will tell whether this will be a more durable response than with just a BCMA CAR. So to conclude, BCMA-directed CAR T therapies are now available for treatment of myeloma. The current indication is in the relapse refractory settings. So you have to have had at least 3 prior lines of therapy. You have to have progressed on a PI, an IMiD, and an anti-CD38 antibody. We're starting to see results of this use of these products in the earlier stages of disease. Obviously, we have CARTITUDE-2 with cohorts A and B that are showing some very impressive results. But here, of course, again, is the longer follow-up that will be important as to whether these can start challenging our current standard of care in the second line or third line. We do now know that KarMMa-3 has met its primary endpoint, and this, I remind you, is a randomized clinical trial of ide-cel versus standard of care. And presumably has met its primary endpoint, which is progression-free survival. So we're hoping that hopefully we will see this data at ASH in December. I think the BCMA therapy is very nicely filled the post-CD38 space. And so I think it's, that's where they fit right now. But I think that they will start challenging sort of even the early lines of therapy in my mind. Unfortunately, relapses continue to occur. So at least at this point, these therapies do not seem curative. And I think the biggest problem still remains issues with making these therapies more accessible to all of our patients, not only even within the United States, but obviously throughout the world. And so with that, I will stop by just showing you sort of the Sarah Cannon team. We are spread out throughout the United States. And so I thank all of my collaborators, and I thank you for your attendance and attention.